The last big topic in chapter 3 is that of transcription and translation. So, transcription is where DNA is coded into mRNA. So, just like a transcriptionist, a medical transcriptionist, their job would be to copy what is said by the doctor and to type it out. So, it literally means the copying of DNA, and it only happens in the nucleus. So it's very important for you to remember that the DNA never leaves the nucleus. Followed by transcription is a process called translation. And that copy of DNA, which is now called mRNA, is decoded and assembled into a polypeptide. And remember, you learned earlier that the ribosome, its function is protein synthesis. The majority of the time, that ribosome is embedded or a part of the endoplasmic reticulum. So the rough endoplasmic reticulum is usually continuous with the nuclear envelope. So the RNA is the go-between molecule that links DNA to proteins. The RNA copies the DNA in the nucleus, and again, it's only in the nucleus, so that's very important to remember. Once it's copied the DNA, it then carries it out into the cytoplasm to the ribosomes. And it travels through the nuclear pore. Again, made up of a protein that's embedded in the nuclear membrane, a double-layered membrane. All RNA is formed in the nucleus. RNA differs from DNA in that uracil is substituted for thymine. So on the test, if I show you a list of nucleotides of bases, you should be able to tell if it's DNA or RNA. So if you see a U, you know it's RNA. If you see a T, you know it's DNA. As you learned in chapter 2, and especially on that macromolecules chart, RNA has 3 different forms. It has messenger RNA, ribosomal RNA, and transfer RNA. The messenger RNA is a copy of the DNA that happens in the nucleus. The ribosomal RNA is the structural material of the ribosomes, which is in the nucleus. And transfer RNA is what is going to transport 2 things. It's going to carry what's called the anticodon and the corresponding amino acid, which is the beginning of that polypeptide sequence. So what happens is we can see the nuclear envelope around the nucleus. The DNA is copied into mRNA. The mRNA then is going to exit through the nuclear pore, and it's going to travel to a ribosome. The ribosome acts like a reader. It reads the mRNA sequence 3 letters at a time. And with every 3 letters, the transfer RNA molecule brings in 2 things. It brings in an anticodon and an amino acid. And those will make more sense on the next couple slides. So now what we're looking at here is translation. There's a lot going on this slide. There's a couple things you don't have to know, but we've already talked about what transcription is. Translation now is the process in which the genetic information from the mRNA is decoded by the ribosome into a polypeptide. So the translators are tRNA molecules, and they recognize the mRNA codon, and they bring in an amino acid. So the kind of the big picture here, simplified version, is the DNA is going to be transcribed into mRNA, and that happens only in the nucleus. So that is transcription. That mRNA molecule is read by the ribosome, 3 letters at a time. And each 3 letters, so every 3 letters are called a codon. So it reads those 3 letters, and then the tRNA molecule brings in what corresponds to that codon. So it has 2 things that it brings in. It has 3 letters, which would be the opposite of the codon. So it's called the anticodon. And that anticodon then corresponds to an amino acid. And you don't need to know what 3 letters correspond to which amino acid or anything like that. If you're curious about it, you can look it up in the appendix in the back of the book. But let's say that the 3 letters are UUU. Then the amino acid might be methionine. So that's all that means. So the tRNA molecule brings 2 things in. So that's the simplified version of what's going to happen. So now in the nucleus, we have the DNA molecule, and it's copied into an mRNA molecule. That mRNA molecule then leaves the nucleus, goes to the ribosome, which is usually on endoplasmic reticulum. This is happening in the cytosol, which remember is the intracellular fluid. So we're still in the cell. So now this ribosome is going to be reading each codon. So the first codon it reads is what's called the start codon. But remember, what you need to know about a codon is that it's 3 letters of mRNA. And since it's RNA, you should know that it has U and not T. So if I had TAG and UAG, you should be able to tell me that this is DNA and this is RNA. So we have RNA here with the U and DNA here with the T. So what happens during translation now? So remember, transcription only happens in the nucleus. Translation happens in the cytoplasm. So the ribosome is reading this 3 letters at a time. You don't need to know the E site, the P site, or the A site. You need to know that a tRNA molecule brings 2 things. It brings the anticodon, which would be these 3 letters, UAC, and it brings an amino acid. And again, you don't have to know the names of amino acids. This just happens to be methionine. There's an enzyme that makes each tRNA molecule. That's not important to know either. But again, the tRNA brings 2 things with it. It has the anticodon, which are these 3 letters, and it brings a corresponding amino acid. You don't need to know initiation, elongation, or translocation. You do need to know once there's one amino acid, it's going to be attached to the next amino acid via a peptide bond. And that's how that polypeptide chain starts to grow. Do remember from chapter 2 that when there's just a string of amino acids, that's called a primary structure. And then that primary structure is going to be folded when it gets to the Golgi apparatus. So that's translation. A simpler and even simpler way to look at transcription and translation is within this DNA molecule. Let's say we have 4 different genes here. These 4 genes are eventually going to produce a protein, and that protein is the polypeptide. So the DNA sequence is separated every 3 letters at a time. Those 3 letters are called a triplet. So you need to know that DNA is made up of triplets, so every 3 letters. So let's say we have TAT as the first triplet. Then that is transcribed into mRNA. Every 3 letter of an mRNA molecule is called a codon. So it's the base sequence here. So if this were our triplet, our codon would be the complementary bases. So remember in chapter 2, I told you GCAT. So you could remember GC is Grand Canyon. You can remember AT is at if you want. It doesn't matter what the memory trick is. So the corresponding nucleotides would be A, U, A. You might have been tempted to say ATA, but remember, if you said ATA, that would be wrong because that would be DNA. AUA is going to be RNA because of uracil. Then, once those codons are read by the ribosome, there's a tRNA molecule. It's sort of like a little bus that floats around that brings 2 things in. It brings in an anticodon plus an amino acid. And again, we don't care what these amino acids are. We just care that it's an amino acid that's brought in. And that's what eventually forms the polypeptide. So let's try to do the last one as a group. You might want to pause this video. I want you to write down what the tRNA would be, and it's an anticodon, and you need to know what 3 nucleotides it would be. Those 3 nucleotides would be U, A, U. And again, because it has U, it is RNA. So these would be the correct sequences from here, from the DNA to the mRNA is transcription. And then translation occurs last, going from here to here. And the tRNA molecule brings in 2 things, an anticodon plus an amino acid. And again, we don't care what amino acid it is.